Good afternoon, everybody, and welcome to the Talking Trial Science webinar. We are very happy to have such a large audience today. First of all, I would like to introduce you my co-chair for today's webinar, Marcel Leis from University of Constance, and our moderator Silke Keller from Tübingen.
Hello, also from my side. I'm Marcel Leis from the University of Constance, and I have the pleasure to introduce this seminar, perhaps because we are a bit closer to Zurich. So we move outside Baden-Württemberg, but not too far. And we have also some direct personal and scientific contacts, so I look especially forward to this. I think for all those that come in more from the outside, I should mention there has been a big MPS, Microphysiological System Congress now in Berlin. The field is really exploding, and Insphero takes a major role and has been doing this for many years. They are probably known with many for liver models, but the field is going on. And the big challenges at the moment are going from an organ model to a disease model, going from something that can produce results to something that can be automated and provide a high throughput. And then, of course, moving from one more organ to multiple organs and organ interactions. And I think these are the three hot areas we will hear something about today. And therefore, I think we all look forward to this.
Yeah, thank you. Indeed, we have two speakers from Insphero. We have Alexandra Title. She's a molecular biologist and she has a strong background in disease pathophysiology. She got a PhD in molecular and translational biomedicine from the Swiss Federal Institute of Technology, and she's in Insphero since 2001, where she's a senior scientist for the islet discovery group. And here she's developing physiologically relevant high-throughput diabetes models. And the second speaker is Lisa Hoelting. She's a biologist and toxicologist. She has got her PhD from University of Constance from the group of professionalized in developmental neurotoxicity and stem cell technology. She has joined Insphero already in 2019, and a focus area is application development of advanced multi-tissue platforms. So thank you both for being here. And before kicking off the talks, we would like to have a quick poll to get to know our audience today. Our first question, we just would like to know if you are working in the 3D in vitro or MPS field. And secondly, if which of these two options you think are the most challenging while working with 3D models. And as third last question, where you think that 3D in vitro MPS have more potential. Yeah, okay, 30% of in vitro biologists. We have also have other, maybe if you want to write in the chat, which is your area of expertise. We also have in vivo biologists, 30%, so it's very interesting. And we have engineer and also end users. Then for the second question, we have a winner. Our participants think that the most challenging part is the in vitro model part. We will hear more maybe in these talks if this is the case. And for the last question, drug development, definitely. She has more points as high potential. The second is probably toxicology. Then it's my pleasure to give the floor to Alex. She will have the first talk of today. Thank you.
Thank you very much. So yeah, those are some interesting results for the polls. I like that we're about half, 30% in vitro, 30% in vivo, so we can have a little battle. And in terms of the answers to the questions, all of the challenges are challenging, basically. So developing a functional in vitro model, is difficult, but also making it scalable and high throughput and everything is also, and reproducible is also quite a challenge as well. So thanks for the introduction, Marcel. You gave a very nice introduction to all of the challenges that we do face in the field and kind of what we're focusing on. So today I'm going to talk to you about human islet microtissues for type 1 and type 2 diabetes modeling and drug discovery. Just to give you a few brief words about Insphero, Marcel already introduced us a little bit, but we started out as a microplate engineering company, and we specialize in making microplates that are suitable for spheroid or microtissue culture. So first we started making the plates, and then we actually started using them. So now we use them to produce microtissues mostly for islet and liver. And we study these in their healthy state, but also we also use them for disease modeling. And we're also heavily involved in technology development. This includes automation, transcriptomics, high content imaging, and biobanking. So as I'm sure many of you have heard, there was the FDA Modernization Act that passed last year. And what this did is it replaced the word animal with non-clinical tests in the law governing the agency's drug assessments. So what this means is that animal tests are no longer a requirement for preclinical tests of a new drug. And this is just a few quotes that are from this article that was published last month in Nature Medicine. The use of animals is expensive and often doesn't reproduce human responses. According to the FDA, over 90% of candidate drugs tested in preclinical trials fail to demonstrate either safety or efficacy when tested in humans. And besides reducing the loss of animal lives, using alternatives can save companies money and time. This means that human in vitro models will have a growing role in drug development. As I said, I'm going to focus on pancreatic islets. So just to make sure we're all on the same page, I'll just give you a brief introduction. So the pancreatic islets are found in the pancreas. They compose the endocrine pancreas, and there are three major cell types: the beta cells that secrete insulin, the alpha cells that secrete glucagon, and the delta cells that secrete somatostatin. And these three major cell types all function together to secrete hormones to keep blood glucose within a very specific range. And it's the dysfunction of pancreatic islets, beta cell dedifferentiation, and beta cell death that leads to dysglycemia and diabetes. And then just very briefly about diabetes. So there are two main types of diabetes, type 1 and type 2 diabetes. And what they have in common is that they all involve a loss of beta cell mass. But the causes of this are different. So type 1 diabetes is an autoimmune disease in which there is an autoimmune attack of the beta cells, and that leads to massive beta cell death, which then results in decreased insulin secretion. And type 2 diabetes is a little more complex. It's basically the culmination of ER and oxidative stress in the beta cells, as well as inflammation that leads to beta cell dysfunction, beta cell death, or beta cell dedifferentiation. And this is usually correlated with obesity and insulin resistance, as well as genetic susceptibility. But as I mentioned, what these two different types of diabetes have in common is that there is a loss of functional beta cell mass. This leads to chronic hyperglycemia if it's left untreated, and there's a need for lifelong insulin therapy in the case of type 1 diabetes, and diabetes therapeutics or even insulin therapy in the case of type 2 diabetes. And this here is some data that shows the beta cell mass of type 1 and type 2 diabetes patients. This is from donor pancreata. And you can see that in the type 1 diabetes patients, you really have nearly a complete loss of beta cells, which is why these patients really don't secrete any insulin anymore. Whereas in type 2 diabetes, it's more moderate. So usually the most you achieve is usually about a 65% loss in beta cell mass. So in the field of diabetes research, there are a lot of mouse models that are used. There's many different models that exist, both to study type 1 and type 2 diabetes. They can be very advantageous because type 2 diabetes especially is multi-organ in nature. So you do have interactions between the adipose tissue, the liver, the brain, and the pancreas. And of course, the ability to generate transgenic mice is also important to study specific genes that are related to diabetes. Some of these models rely on non-physiological sources of diabetes induction. So for example, chemical ablation of the beta cells, for example, is something called STZ. And some of the models are a little bit more physiological. Sometimes the mice are put on a high-fat diet, which is supposed to mimic the human condition. Many of the results that are found in mouse models, though, are not translatable. And there's several different reasons for this. So certain mouse strains are actually more susceptible to diabetes than others. So depending on the mouse model that you use, you might get very different results. The dose of a new drug can be challenging to translate to human. Female mice are more resistant to type 1 and type 2 diabetes modeling in mice. And so they're often excluded from in vivo studies. But then, of course, this reduces the translatability to the human population. And there's also inherent differences between the species. So beta cells in mice have a higher proliferative capacity compared to human beta cells. The structure of the islets is different, and the glycemic set point is actually different between mouse and human as well. So our basal glucose levels are actually quite different. And another important point is that the age at which mice are studied also tends to be a lot younger than the humans who would receive a diabetes treatment. And that's especially relevant for proliferation studies, for example, since proliferation rate of beta cells decreases with age. So these are all good reasons to focus on human islet research, but there's some challenges associated with that. So human beta cell lines in general tend to not be very translatable. This is due to several reasons. They are only beta cells, so you do lack the alpha and the delta cells, which are important for islet function. And in general, they just don't reproduce the proliferative rate and stress resistance of an in vivo system. So the best gold standard for islet biology is to use isolated islets from donors. But as you can see, this presents quite a few challenges. So in the middle here, we have a staining of islets that were isolated from a human donor. And you can see that there's really quite a lot of variation between the islets in terms of their composition. So every color is a different hormone, insulin, glucagon, and somatostatin, which represent the different cell types. So different islets have different proportions of the cell types. There's different sizes, and there's also exocrine contaminants, which can also interfere with the function of the islets. So what this means is that when one wants to work with native human islets, they have to be pooled together, typically in pools of 20 to 30 islets per well. And this is typically done in a 12 well, 24 well plate, which makes them incompatible with screening. And then another important limitation of native islets is that they have a short viability in culture, so they can only be kept for about 3 to 4 days. Another way to work with the native human islets is to disperse them into 2D onto a cell culture surface. Again, there we lack 3D cell-to-cell interactions, which are important for islet functionality, and the cell culture surface can actually have an interference on the function as well. And finally, patient islets from type 1 and type 2 diabetes patients are not readily available. So there is a need to have a model that can be conducive to disease modeling. And that typically would require a longer culture period than the 3 to 4 days allowed by the native islets. So what we do is we make something that we call human islet microtissues. So we take the isolated native human islets from donors and we dissociate them into single cells and reaggregate them into these microtissues. And we do this using a hanging drop system that we established here. And it basically leads to the final product of having a 96 well plate with 1 microtissue per well. And each microtissue is homogeneous in size and also a lot more homogeneous in composition compared to the original islet prep. And another important thing is that these islet microtissues can be cultured for over 28 days. And this is just because of their smaller size and their increased purity compared to the original islets. So this is very important for drug testing, for looking at long-term effects of different drugs in terms of toxicity or in terms of functionality. So one interesting thing about islets is that their structure actually varies really quite a bit between species. So you can see on this phylogenetic tree that different types of animals have a very different ratio of beta and alpha cells, but also different organization of the islets themselves. And this actually has an effect on the functionality of the islets as it affects the communication between the different cell types. And what's interesting is when we make microtissues from rodent pancreas, so we actually reproduce, and from human, we actually reproduce the structure of the original species. So the rodent pancreas has a lot more beta cells that are in the core, and then you have a alpha cell rich mantle, and that's reproduced in the rat islet microtissues, whereas the human pancreas has these sheaths of glucagon positive cells that are kind of intermingled among the beta cells that are in green here, and that's also maintained in the human islet microtissues. And again, this is just aggregation in a hanging drop, so this just happens naturally. But as I mentioned, it is important for the functionality of the microtissues as the cell-to-cell communications are key. Another interesting observation that we've made is that there's really quite a lot of human variation in insulin secretion. So here we have plotted 172 donors. So we've been doing this for a while here. So this is 172 different human donors for which we've generated human islet microtissues and then looked at basal insulin secretion at 2.8 millimolar glucose concentration, stimulated insulin secretion at 16.7 millimolar glucose, and this last graph is just the fold change of the two. But you can see that there's quite a big spread in the data, which means that there's a big spread in functionality of islets in the human population, and that's not necessarily something that would be captured in a mouse experiment with an inbred mouse line, for example. So now I'm going to tell you a little bit about the different applications that we can use these microtissues for. So as I mentioned, type 1 diabetes is a autoimmune disease. So one way to model type 1 diabetes in vitro is to use cytokines. So here we take the microtissues that we produce and culture them in the presence of 3 cytokines that are relevant for type 1 diabetes. This is interferon gamma, IL-1 beta, and TNF alpha. And so we just culture the islets in the presence of these cytokines and then perform functional analysis. So you can see here that with the lowest concentrations of the cytokine cocktail, we already do see some effects on insulin and glucagon secretion. So this is a representation of stress. And there's also a direct effect on functionality as well. So as we increase the concentration of the cytokine cocktail, we have a decrease in stimulated insulin secretion, meaning the beta cells can't do their job as well as they normally should. And we also have a loss in intracellular insulin content. So this is the pool of readily available insulin that should be available to be secreted in response to a glucose load. If we look at caspase 3-7 activity, we also see an increase as you increase the cytokine concentration. So these cells are dying very much as is the case in type 1 diabetes. So that's kind of the more basic type 1 model, but we can also do islet microtissue immune cell co-cultures. So we do this just in our same Insphero plates where we have the microtissues that are being cultured. So we basically co-culture the microtissues with PBMCs either in their naive state or with PBMCs that have been activated with CD3, CD28, which basically induces a broad activation of the PBMCs. And as you can see in these little bright field pictures here, the naive PBMCs don't do much, whereas the activated PBMCs do latch on to the microtissue and destroy it. So that's a little bit more of a general model. And then we also have another model where we do co-culture with CTLs that are specific for pre-proinsulin. So pre-proinsulin is highly expressed in beta cells. It's the precursor to insulin, actually. And this is actually one of the most common autoantigens in type 1 diabetes patients. And so if we co-culture the islet microtissues with these PPI CTLs, then we also have an attack. And this has a direct effect on the functionality of the islets. So in the bottom graph here, in green on the left side is the PBMCs that are naive, and in purple we have the activated PBMCs. And as you can see, as you increase the number of activated PBMCs that are in co-culture with the human islet microtissues, you have a big drop in stimulated insulin secretion as well as in intracellular insulin content. And there's also a similar case for the CTL PPI co-culture. So we have a drop in stimulated and total insulin content as well. And this is some live imaging of the PBMC islet microtissue co-culture. So in green, we've dyed the islets, in green, we've dyed the islets, and the PBMCs are dyed with red. And as you can see, the naive PBMCs in the middle really don't do much. They just kind of float around, but don't bother the islet microtissue too much. Whereas on the right side, the activated PBMCs really do go in and attack the islet microtissue. So you can imagine that, this type of tool, whether it's live imaging or just microscopy from fixed tissues, can be pretty important to look at infiltration of immune cells into the islet microtissue and to judge, the potential of a new drug to prevent infiltration of T cells, for example. So this is just a couple of examples of how we can use these type 1 diabetes models for drug efficacy studies. So in this particular case, this was published last year in Clinical Immunology, we were interested in looking at a drug that's actually used commonly for type 2 diabetes called liraglutide, and to see if it had anti-inflammatory effects in the context of type 1 diabetes. And so we initiated a compound treatment with liraglutide on day 0, so basically 1 day before the cytokine treatment. And as you can see, in the absence of cytokines, that would be the C0 concentration here, we have a big increase in stimulated insulin secretion. So that is the effect directly on the beta cells that you'd expect from such as in a type 2 diabetes treatment case. But what's important is that the increased stimulated insulin secretion is also maintained in the presence of cytokines. So obviously not to as much, not as, well, there's still a decline in function, but there is still higher function in the islets that have been treated with the highest dose of liraglutide compared to no liraglutide. And we also did a similar treatment in the PBMC islet co-culture, where we pre-treated the islet microtissues for 1 day with liraglutide and then kept it in during the co-culture as well with the PBMCs. And this is again similar, where you can see that there is an increase in stimulated insulin secretion in the presence of liraglutide relative to the case where there is no liraglutide. And also, this is then results from microscopy data. There was actually reduced T cell infiltration into the microtissues. So we can quantify that using microscopy. And something that's not shown here is that the liraglutide actually had a direct effect on the cytokine secretion of the PBMCs. So it really does have an anti-inflammatory effect. We can also perform type 2 diabetes modeling. We do this with using glucotoxicity and glucolipotoxicity models. So what we do here is we take the microtissues and culture them for 1 to 2 weeks in the presence of high glucose and free fatty acids. And so in the top row here, we have a staining of 5.5 millimolar glucose. So this is the standard condition. And you can see that there's high expression of insulin and high expression of glucagon, and also of amylin, which is typically co-secreted with insulin. But when we culture the islet microtissues with high glucose, 16.7 millimolar, and free fatty acids, you have a strong reduction in insulin expression and actually in glucagon expression as well. And you also have the formation of amyloid plaques, which is something that also occurs in type 2 diabetes patients. And if we look at the functionality, we can see that there's also reduction in secreted insulin in the presence of high glucose. Actually, already without free fatty acids, the free fatty acids have the strongest effect at the intermediate glucose concentrations here, so at 8 millimolar. And then there's also a strong decline at the high glucose and free fatty acids concentrations. And we also see a similar story here for the total insulin content as well. And again, these types of models can be used for identifying new drugs or testing drug efficacy. So here there was a small screen that was conducted. And what we did is, so we used this model to identify compounds that would rescue the effect of the glucolipotox treatment. And so you can see that this drug, which is blinded, leads to a strong increase in stimulated insulin secretion, actually even higher than the control, even in the presence of glucolipotox media, and the insulin content is at least partially rescued. So just a couple more applications I want to mention. So we can perform viral transduction in the microtissues as well. This is important because, one advantage of mouse models is that you can do transgenics, and human islets in particular are hard to manipulate from the gene expression of them because they're a 3D structure. So you can't really do efficient viral transduction of a 3D structure. But since we make our microtissues by including a single cell phase, that actually allows us to manipulate gene expression as well. So here we did a test where we transduced the islets before reaggregation, during reaggregation, or after reaggregation with adenovirus. And this after reaggregation would be the equivalent of trying to transduce a native isolated human islet. And not surprisingly, you have a lot higher efficiency of transduction if you do it during the single cell phase, so before or during reaggregation, and basically no transduction after the reaggregation. So transduction can be used then to overexpress a gene. And we've also had some cases where we were able to knock down gene expression by doing AAV-driven shRNA expression. This is again a gene that was blinded since it was from a customer project, but we got 80% knockdown of the gene in the whole islet. So this is very powerful in terms of, basically performing basic islet biology as well. And then just one more example, this is the example of proliferation. Since, as I mentioned, type 1 and type 2 diabetes both involve a loss of beta cell mass. And as you can see on the graph here, one of the problems is that human adult beta cells basically don't proliferate. So in the neonatal phase, you have a certain amount of proliferation, but then that declines with age, and it continues to decline to very low levels as we get even older. And this means that those beta cells that are lost due to whatever attack can no longer be replenished. So in the field, there's a lot of interest in identifying factors that could induce proliferation of beta cells. So this is a list that my colleague prepared a couple years ago. It's probably a little bit out of date, but it shows the molecules that induce beta cell replication in rodents and other eukaryotes. And this is that same list in terms of how effective or which molecules are actually effective in humans. So proliferation is an example of something that does make a lot of sense to study directly in humans, just to have actual translatability of the results. So we created a platform where we take the islet microtissues and we treat them with a compound, putative proliferative compound for up to 21 days. And we include EDU incorporation in the final 4 days of the treatment. And then we perform staining and imaging in 3D, and then quantification is in 3D as well. And then we can also use parallel plates for functional assessment to make sure that the cells that are proliferating are actually still functional. And we use this for a pilot study, just to show an example, where we used harmine, which is a well-known inducer of islet cell proliferation, in increasing doses. And as you can see, we do see increased proliferation as we increase the doses. And this can be quantified in 3D throughout the whole microtissue. So in summary, 3D in vitro models are more physiologically relevant than traditional 2D in vitro models, especially in the islet field, since 3D cellular interactions are crucial for proper islet function. Human 3D in vitro islet models enable the earlier study of potential new diabetes drugs in a reproducible and highly relevant human system. Gene expression manipulation and the study of specific disease-relevant mutations is also possible in human islets using viral transduction and CRISPR. And while certain complexities of diabetes can only be studied in mouse models due to the complex multi-organ nature of the disease, complex microphysiological systems may actually bridge this gap. And that's something that Lisa will tell you a little bit more about. Thank you very much for listening.
Thank you, Alex. And I directly pass the microphone to Lisa to continue.
And then also welcome from my side. I will now continue, and I'd like to give you some insight in our latest R&D activities with a focus on multi-tissue platforms. From Alex, you have heard now a lot about our pancreatic islet model and how we can use it as a drug discovery tool or to investigate new molecular mechanism. And beside our pancreatic islet model, we also have 3D tumors and 3D liver models that can be used for drug efficacy testing and safety assessment. And hopefully soon, we also have a adipose model that can be used for similar application. So as you can see already, with focusing only on one single tissue in a 96 or 384 well plate, you can answer a lot of questions. However, if you are more interested in studying tissue-tissue crosstalk, for example, how the disruption of one tissue affect the homeostasis of other tissues, then of course you need an in vitro system that reflect this crosstalk. Or another example, if you are interested in how PBMC have an effect on tissue homeostasis or influence drug efficacy or disease progression, then of course you need an in vitro system that enables the addition of PBMCs. And I would like to show you now the approach that we take to model tissue-tissue crosstalk in vitro. So what we want to add is we want to add physiological flow condition. We want to have the presence of immune cells. And of course, we want to enable tissue-tissue interaction. This, I would say, requirements more on the biological side, but while we are adding this new level of complexity to our existing models, we have also some requirements on the technical side. So we want to prevent the scalability, which means that the new platform should be compatible with automation. Otherwise, it's not possible for scaling it up. And we want to have a robust handling, which means that the new the design of the new platform is chosen in a way that can keep the operational complexity very low, and by this avoid microtissue loss. And of course, we want to have that the new platform is also compatible with all the endpoint assays that we have already developed for our standard plates, starting from biochemical assays over high content imaging to omic approach like transcriptomics. And as you can imagine, to fulfill all the requirements, this is not a process that's taken place overnight. And I think this is nicely reflected with this in this timeline. So the first approaches have been taken more than 10 years ago by our VP Olivier Frey, who was at this time a postdoc at the ETH, and it took some years from the initial trials and concept to get an automated and industry compatible platform. So this is our Acura flow chip, and we did some further development during the last years, and we came up this year with 3 new platforms. And the Acura Flow 384 version is actually the direct followers of the Acura Flow chip, so the transformation from a chip format to a plate format. And you now may ask a question, why 3 platforms and not only 1? So the answer is that 2 years ago, we got a request from the industry to develop an assay that can study or that can detect immune-mediated liver toxicity by bispecific antibodies. And then we thought, okay, so for this specific application and for many others application, you only need 2 tissues. You need a tumor and you need a liver, and the addition of PBMC. So maybe a multi-tissue platform where you can culture up to 12 microtissues might be a little bit too complex. And so we want to reduce the complexity, and by this we can increase the scalability. And so we came up with the Acura twin plate. So this is based on a 3/8 well plate, always connecting 2 wells with a little channel. And so you can place the tumor in one and the liver spheroid in the second well. And then you can co-culture them with PBMCs, and then you can assess on target off-target toxicity simultaneously. And the second or the third plate format is the immune flow chip. So the reason why we developed the immune flow chip was that we realized if we now put immune cells in the Acura flow chip, immune cells quickly start to sediment and this would clog the channel. So if we now want to study how flow affect the immune cell population or how immune cell attach to a tissue, then of course we need a platform where we can co-culture it with multi-tissues and where we can keep the cells in suspension for several days. And so we came up with the immune flow chip. Yeah, 3 different platforms with different features for specific applications. And I will now go and tell you a little bit more about the Acura Flow platform. So this is our multi-tissue platform. It's based again on a 3/8 well format, but you can see this is the bottom view. The bottom has on the bottom you can see the channel structure, and we have always 12 compartments that are interconnected with a thin channel, and on each side there is a reservoir. And the flow is induced by gravity force, which means that we put the plate on a tilting device, which is the size of a little shoebox. So you can easily put it into the incubator. And by tilting back and forth, the liquid flows from the upper reservoir along the channel, interconnecting the microtissues in the individual compartments to the lower reservoir, back and forth. And since the bottom is transparent, it's made of polystyrene, it's also image compatible. And of course, you can exchange the media all the time or just sample media for some further analysis. And also very important is to remove that you can remove the tissue afterwards. And now I'd like to show you how you can use such a multi-tissue platform to study liver islet crosstalk. Here again, some background information, but Alex also already nicely explained it. So liver and islet are the key regulators or responsible in keeping our blood glucose level in a very tiny range. So if the blood glucose level is very low, glucagon is secreted by the alpha cells, And this induced hepatic glucose production in the liver. So glucose is released into the bloodstream to increase the blood glucose level, and the opposite way. So if the blood glucose level is very high, then insulin is secreted by the beta cells, which inhibits the hepatic glucose production and also induce glucose uptake by the peripheral organs. And this then let the blood glucose level decrease again. And the first thing that we have done is that we have characterized our liver, if the liver is our liver model, if it's responsible to glucagon and insulin. And so as you can see here, we have recapitulated the feeding state and measured the glucose release at the presence of different insulin concentrations. And you can see that with increasing insulin concentration, the glucose release is decreased. And also the other way around. So we have recapitulated the fasting state by showing that with increasing concentration of glucagon, the glucose release can be increased. And here, to complete the picture, we also did with the islet, and of course, glucose-induced insulin secretion, and on the other hand, simultaneously inhibit the glucagon secretion by the islet cells. So based on that, we decided that our liver and islet models are suitable to put them together on the chip and do some co-culture experiments to test the crosstalk between these two organs. And the way how we are doing it, I call it an off-chip production, on-chip integration, which means that we produce the islet and liver microtissues according to their standard protocols in this 96 or 384 well plate. And then at the given time point, after we have done our QC, we can transfer the tissues just by picking them up and doing a contact transfer and transfer them into the Acura flow chip. Of course, you can do it manually, one by one, or what we have done is to increase a little bit the speed. We have developed some protocols to do it semi-automated with via flow. So here you see in picture how it looks like if you do a parallel microtissue transfer from a 384 plate into the Acura flow plate. And if you now may wonder what is the adapter, we created because what we realized that the compartments are very nicely aligned. However, the pipette tips are not that good aligned. So we built up an adapter, put them on top of our Acura flow plate, and there are holes at the defined position, and the holes somehow guide the pipette tip to find the right location, so that we really target the compartment and the microtissue falls into the compartment. Okay, so the results I'm showing here now, they were made on the previous version. So that's the reason why you only see 10 compartments. And this is now a scan of 8 flow channels. And in this example, we have co-cultured always 8 liver microtissue with 2 islet microtissues under physiological glucose concentration for a time of 7 days. And then we looked for islet as well as liver-specific endpoints. So here you can see that the chronic insulin secretion over time is in the co-culture is similar to the insulin secretion in the islet-only culture. However, if we have a look at the glucose-stimulated insulin secretion, which we performed at day 7, you see that in the co-culture, the islet microtissues are much more responsive than in the only islet culture. On the liver side, we see that the lipid content that we measured is quite similar between both conditions, so the co and the mono culture. However, if we now challenge the system with a high glucose diet or a high glucose plus free fatty acid diet, we see on the one hand that the insulin secretion over time increased as expected. maybe a little bit higher in the co-culture than in the monoculture. And that, as expected, we see that we have a dysfunctional GSIS in both conditions. If we now have a look at the liver and checked again the lipid content in the liver, we see that with increasing concentrations of the insulin, we also could measure an increase in the lipid in the liver that we don't see if we have only mono liver cultures. So I think this nice data that characterized the system, and I think now it's ready for the first compound test. Or maybe also what we would like to test next is how, as I mentioned before, we're also developing the adipose model. So how the endocrine function of the adipose model affects the liver islet crosstalk. And with that, I would like to make a little announcement at the end. So we are currently running an initiative with the physician committee, and this is organizations that support human-specific non-animal approaches. And I think it might be of interest for all academic as well as non-profit organization to apply for a grant that they are offering worth 20K and can be used for our Insphero technologies to support human-based in vitro methods. You simply have to go to the website, download the application form and send it together with a CV to this address. And deadline is the 23rd of August. And with that, I'm at the end of my presentation and I'm looking forward for the discussion.
Well, thank you, Alex and Lisa. I think this was very lively, very instructive. So it gives people a good chance to ask what now or why and how. So let's see, perhaps, Georgia, you have an overview whether there's written questions already?
Yeah, maybe I can start, let's say the first one. It's, are you doing something with nervous system tissues? So first, this talk was focused on other organs, but is the technology already applied in other?
Maybe I can answer this question. So we have some little project developing neurospheres. So there is a little project that is going on.
All right, then we have another question. It says, what is the viability of your tissues in your chip model systems?
So the data that I've shown were for 7 days. We also did experiments for 14 days and they were quite viable, but actually I've never tested longer periods, but should be work as well. So
I was just going to comment, maybe you can comment, Lisa, on how you do the media exchange or how often you do the media exchange, for example.
Yeah, so I mean, it also depends on the experiment. So we usually do the medium exchange every 2 to 3 days. And I think the liver and the islet microtissues are viable or functional, active for 28 days. So I would guess that also we can keep them for this time period in the chip.
Okay, then maybe a question. And it's about the ratio of the co-culture, of the 2 cell types for the co-culture. So in the older question is, especially in the liver and the islet culture, you showed that there were more liver spheroids and less islet spheroids.
Mm-hmm. So we did some previous, so it's based on some previous experiments that we have done. It's actually what we are looking for, about the functionality. So depending on the endpoint you're looking for, you maybe have to change the ratio. And I think now we will also do some further experiments with the new version, because there we have another volume of media, which also could affect the crosstalk between the 2 tissues.
All right, then we have another question from a biomedical engineering and medical physics student from Munich. And he or she would like to know if Insphero also offers positions for an internship for 1 year or any student-related positions. So maybe you could comment on that as well.
Yeah, we've had quite a few master's students, actually. We have a few right now. So yeah, if there's some interest, send an email. We have to see the capacity, of course, but it's something that we're interested in, yeah.
All right. Would you maybe share an email address in the chat so he or she can maybe directly send you a message?
Here we go for next. Then it's well, it's a comment. We talk. It's very exciting to see how advanced these models are, and then thinking of the implementation of these models more widely on the regulatory side. Has any data generated with this platform be submitted already to any regulatory agency? And when do you think these models could be ready to reduce or replacing animal models?
I mean, maybe, so we have quite a big liver toxicology team. So I think that's probably the platform that's been the most involved in regulatory cases so far. I don't know for sure that, I mean, I do believe that it has probably been used, right, Lisa, for
I think there are some discussion ongoing now. Yeah.
I mean, on our team, we have had to prepare some FDA documentations before for some of the service work we do. So I'm not sure, we don't really know the full story sometimes. So I assume the customer submitted that somewhere, but we have filled out those documents before as well.
And then there is one question going into a similar direction. Can you use the liver islet models for drug testing already, or is it still in the development phase?
Yeah, so I think what I have shown now was basic characterization, and it would be now good to do some compound test and to define a little bit more the application and to define the endpoints. Yeah, so we are on the way to it.
Okay,
perhaps Lisa, I can follow up. Perhaps some are interested. So this is like, is it ready for you? And then like, what would it mean? Like, are the plates being sold? Could others set up a system like this? Drug testing can be done at many different levels, like for, FDA registration or for general pharmacological interest.
Yeah, so actually we are open to give or to sell the platform, and so that also, I mean, it's a nice platform, and I think there are a lot of opportunities, and it needs to be explored, and so we can give it away, and we are also open for collaboration and also for paid R&D to bring it a little bit further.
Okay, then maybe the last question, let's see, is how have you done other characterization beside looking at the insulin secretion, and if you can state how close they are to animal models?
How close they are to animal models is a good question, because we don't really do the comparison to animal models. So I can't really answer that. Maybe another question that's relevant is how close they are to the human actual in vivo situation. And that we, in terms of the structure, they look very similar. They basically function like really good human islets, because we make them small so that they can survive longer. So there's no core necrosis. So the bigger islets from just an isolation do tend to develop necrosis over time. So we keep ours small, and actually small human islets do tend to function a lot better than the bigger ones. So our microtissues are kind of like small human islets. And in terms of the fold secretion, we actually get really high stimulation, probably because of this small size and because of the increased purity. So when we do the reaggregation, we actually exclude the exocrine tissue, which is naturally present in the isolation, and that because the exocrine tissue does seem to have an effect on secretion. And so we end up with a more pure islet, which secretes very well. So maybe that's a comment to the human comparison. And I forgot the beginning of the question. You had... The other endpoints. I mean, yeah, so we mostly focus on beta cells. So the beta cells are the biggest interest for diabetes research. So that's insulin secretion, but we also look at glucagon secretion, for example. We also look at things like amyloid deposition, which I showed in one of the slides. Yeah, I would say function is the biggest one. I mean, proliferative capacity is something else. Yeah, survival, things like that.
I try to read another aspect in this question. I think one is if you have the multi-organ platform, you have to exchange through the flow, but then there could be also different cell types within an organ. You know, like in the liver, there could be like 3 or 4 different cell types, or in the brain, there could be glia and neurons. So what do you say about the study of such interactions?
So what we are usually doing, so we have the, for the liver model, we usually use a co-culture, so hepatocytes with NPCs. And I think it's also possible, so we have the NASH model where we have adding stellate cells, and this might also change then the outcome. So it depends on the question. Yeah, but I think the chips that I presented is not limited to liver or islet. So actually, as a requirement for the tissues that you can add into, it has to be transferable. So the tissue has to be compact so that you can transfer it from one plate to the other. And the size, it should not be bigger than 600 micrometers in diameter, because the structure of the compartment is only 800 micrometers. So it's also open for totally other fields. But since we are focusing here on liver and islet, we also have done a lot of studies with liver and tumor. So yeah, I think it's one of the papers that Georgia now shared the link to, is about the bioactivation of some chemotherapeuticals, which have been done in their CoaFlow chip.
So you have a liver as bioactivator, and then you have the tumor.
Yeah.
Okay, I think we have a bit run out of time. Really was really interesting, and there were other questions indeed coming, but we will then we can follow up in another talk. Thank you all our participants and our speakers, and to the moderators Marcel and Silke. We will see you again next month for the next webinar. Thank you all.
Yeah.
Bye bye.
Thank you.
Thank you. Bye bye.
Bye.